Okay, good morning guys. So welcome to the 9th day of this Gian course, which is on next generation sustainable alternative proteins for food. Am I audible to you all guys? Okay. So I just want to quickly take you through that what we do here at NIT Rourkela, and then we will go through what exactly we have to study today. So our group mostly emphasizes research related to plant proteins, and then we also emphasize this research related to starch, and then we use the combinations of plant proteins and starch in order to do the delivery of bio compounds. So in plant proteins we have been largely targeting the exploration of alternative proteins, or by alternative proteins we mean, for instance, plant proteins also, or dairy proteins also is an alternative protein, but largely the global scenario is that most of the people in the world, or scientists or academicians, industrialists or R&D centers, they are exploring different kinds of proteins for meeting the global demands. So alternative proteins generally means plant proteins. Also we are into modification of different kinds of properties of plant proteins, because plant proteins as such cannot be so much tailored for different kinds of food applications. So therefore we have to modify them, and we use different strategies in our research group to tailor the properties of plant proteins. And we recently have started also some work related to plant based meat analogs. We have got a very good project, and we will be starting that very soon. And this non-conventional starch sources has been also predominantly an area of research in our group, and we are exploring a lot of non-conventional starch. So recent one is using millet based starch for different kinds of properties, and then similarly we also do starch modification processes in our research group. So one of our PhD students is working on plant proteins, another one is working on starch, and then the combinations of plant proteins and starch we are exploring together with master students, PhD students, postdocs and everything. So that is kind of an idea about what we are doing in our lab, and we have published some very fine papers related to plant proteins also. We have published some of the very good papers related to starch also, and delivery of bio compounds in some of the very finest journals in the field. So if you want to explore more about that, we will take you through. So today, I mean, in the past lectures you have read a lot about plant proteins. So then maybe you should have also a fair idea of proteins as such, different kinds of properties associated with proteins and different kinds of properties associated with plant proteins. And then you should also have a fair idea of what are the different kinds of properties that can be tailored, or for example when we do modification of proteins, so what kind of properties we change. For example, we change functional properties, we change physicochemical properties, we change rheological properties, or we change surface hydrophobicity. A lot of things are into play when it comes to plant proteins. So but we largely want to emphasize today about the property of plant proteins, which is referred to as astringency. And astringency as such we will discuss in the coming slides. So but when we want to understand astringency, we will be largely discussing about two important properties. One is the rheology and another is tribology. So I understand that most of the people here are in the starting of their master's program, so probably you might not have kind of a fair knowledge of these two properties, but I will try to start from very basic ideas to some of the higher knowledge about these two properties. And the tribology, we are proud to be the only institution in India which is working on tribology of plant proteins. We have the equipment to work on tribology, and I have worked on tribology in University of Leeds also. Okay, so when we talk about plant proteins, as I said that when we are exploring different kinds of proteins, alternative proteins to be specific, so plant proteins are at the front runners of alternative proteins. So in the past, people tried to study, for instance, switching over from meat based products to dairy based alternatives, and then starting with soy proteins and blah 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 blah, and then as of now we are mostly focusing on alternative proteins. So alternative proteins, for example, leaf proteins, or somebody is studying, say for instance, legume proteins also, cereal based proteins also. So they all come under the umbrella of these plant protein systems. And also in the introductory lectures of this Gian course, you would have been taken through that why we are, or the global shift is changing from animal based products to plant based products. So there are a number of reasons. One of the reasons is growing population. So we are expecting roughly around 9.5 billion to 10 billion population by around 2050. So that is one of the concerns that we as human beings on this globe are facing. So the enormous amount of population that will be living on the globe, and then day by day due to industrialization, due to urbanization, the land that is feeding us, the agricultural land that is feeding us is shrinking day by day. So the agricultural land is decreasing. So that means in order to sustain the global population, we have to switch over from traditional systems to so we have to shift from conventional food. For example, one of our conventional food is, say for example, rice, or we're consuming other types of flours, and we are consuming, say for example, chicken, we are containing mutton or whatever be. So probably in the coming years there will be shortage of all these products. So therefore the one of the challenge that the global population is facing, that we need to have a sustainable source that will be helping us to meet our daily requirements. So therefore one of the alternatives to all this, one of the solutions to all this is plant based foods. So because number one, they are environmentally safe, they don't cause so much harm to environment. Second, they are healthy. So plant based meals as such, if we just don't talk about plant based proteins, if we talk about plant based food only, they have lesser health concerns than animal based foods. So I am not educating here that we should 100% switch over from animal based food to plant based food, but generally it has been seen that the health concerns associated with meat, for example, or any other this animal based foods are higher in number than those associated with plant based foods. For example, animal based foods, they largely come with problems such as coronary heart disease or CVDs. A lot of problems are associated with animal based food. So therefore the environmental aspects, the healthcare or ethical concerns of some of the people in the world, because of religion or because of their own concerns, they don't eat meat, or they want, for example, certain kind of meat, for example halal meat, kosher meat, a lot of stuff around that. So the plant based stuff, plant based products do not have those kind of associated problems. Neither they come with environmental concerns, neither they come with health concerns, neither they come with ethical reasons. Also, we have seen, this is again an environmental aspect, that animal based stuff, whenever we have to generate animal based food, whether that is meat, whether that is eggs, or whether that contains there's dairy, it comes with lot of greenhouse emissions. So the greenhouse emissions are generally double when we generate animal based foods contrary to or compared to plant based foods. So but one of the problems, I mean, we largely see in all of you who want to study plant proteins in their research, or maybe go to industries and work with plant proteins, it is not as easy as we assume it to be. Like the shift from animal based foods to plant based foods is not as easy as we assume it to be. Like we go to the labs and we isolate plant proteins, say for example we isolate it from soy proteins, we isolate it from bean proteins, we isolate whey proteins, or any other plant based or any other alternatives, whatever be. So it is not as easy as we assume it to be, that the shift from animal based foods to plant based foods is not that easy. Number one, so most of the people are not willing to do that. Why should I shift from plant based food? I am good with animal based foods. Number one of the easiest kind of say people not being so welcoming to plant proteins, or they don't want to consume plant proteins, is that, for example, animal proteins taste better. Meat tastes better than, for example, say lentils, although we have proteins here, proteins there, but it's just about taste. So people do not generally adopt this. Other scientifically, plant-based proteins have poor sensory aspects. So when we taste plant-based proteins, they, as I said, they are not tasty as animal-based proteins are. So there are a lot of reasons to that. One of the reasons is, for example, they have plant-based proteins come with very high surface hydrophobicity. They have low water binding capacities. They have low oil binding capacities, and therefore the performances of these plant-based proteins in different food systems is very limited. So when we want to, for example, incorporate these plant-based proteins into different kinds of systems, for example, emulsions, for example, yogurts or other foods, so they do not function the way animal-based proteins function. So if you incorporate whey proteins in, say, for example, smoothies, they will behave better than incorporating, for example, pea proteins in smoothies. Okay, so we will just go through all this in the coming slides. Also, what I want to emphasize here is more this one. So this one I want to emphasize more here, and you will keep it as a note: astringency. So whenever we talk about plant proteins, this is one of the major hurdles. This is one of the major hurdles that most of the scientists in the world do not take care of right now. Most of the people who are researching on plant proteins, they just randomly go isolate plant proteins and say that our plant proteins is the best. Soy proteins, for example, leaf proteins, for example, bean proteins, ABCD, but the associated problems are always negated. I mean, we don't take care of all those associated problems. So one of the major drawbacks with commercialization or adoption of plant proteins is that plant proteins come with astringency, or they come with dry sensations. So whenever we consume plant proteins, I'm not sure how many of you have consumed plant proteins as such. If you have consumed, for example, this snack bars, or for example chocolate bars laid with lot of plant proteins, you will see that your mouth is always dry. Okay, so we will draw the parallels to understand how this happens. So as of now, just to lay the floor, just to make you understand that plant proteins come with lot of astringency or dry mouth feeling or puckering feeling. Puckering feeling means kind of a sandy texture on your tongue. Okay, we will go through all this one by one. So again here, then we conclude from the first slide that problems aside or the way outs aside, one of the major drawbacks with commercialization or adoption of plant proteins is that this is a serious bottleneck. So that the bottleneck means you have to pass certain states, and those states are choked. So that means it's very difficult to pass through certain barriers. So those barriers are called as bottlenecks. So the barrier to this adoption of plant proteins is that astringency is one of the major key factors that has to be taken care of. All the researchers who are into plant proteins, they have to study astringency also. Okay, so majority of the people who are researchers, or majority of the people who are students, or majority of the people who might be R&D experts, so they largely do not take into consideration the astringency effects. Okay, so we have seen there are a number of substances in the world. Not just plant proteins. There are a number of substances in the world, not just plant proteins, that come with off flavors, or that come with bitterness, or that come with beany flavors. If you have ever consumed soy based milk, it contains off flavor. It has a beany flavor. Okay, so if you consume, for example, something made from paste of beans, which is traditionally consumed, a beany paste incorporated in buns, the way we have KFC burgers here laid with potato, what do we call that? KFC burgers. Yeah, for example, KFC burgers laid with, for example, potato based patties or chicken based patties. In China, the traditional, I mean, kind of a sweet or traditional meal which they consume, which is parallel to KFC burgers, is that they have these buns filled with beany pastes. So if you consume that also, you will feel that it is dry in nature, it has off flavors. So if we taste wine also, if we taste green tea also, you will also feel the pungency, or you will also feel the astringency, or you will also feel that dryness in your mouth. So now whether that is less, whether that is more, that depends. So we will go through in the coming slides that how exactly it is formed. So, what is the way forward, and then we will relate all these things to our plant based foods. Okay. Or meanwhile, if you have any questions in between also, so please feel free to ask that. Okay. So, one of the prominent scientists in the world, the known figure in the world who has started pioneering work on this is Professor Sarkar. She is the professor at University of Leeds. I had the privilege of working with her, and that is how I started working on plant proteins and astringency processes. Okay, plant proteins and astringency processes, pioneer in the world at University of Leeds. Also, there is one more professor, his name is Gian. You can SHE, Gian Chen. He University of Singapore. And we have one more this professor. His name is Glenn Yakubo. So he was previously at a different university. Has also moved to University of Leeds now. So these are three prominent figures in the world who are working on plant proteins and associating plant protein. Okay. So if we concentrate plant sourced proteins, or we concentrate based foods. So if we just concentrate, for example, seed, then we concentrate on nuts, then we concentrate on tubers, then we concentrate on oil seeds, then we concentrate on legumes. All these substances taken together, we can get proteins from all these substances. So we do not only get proteins from legumes, for example. We do not get proteins from seeds. Proteins from substances, all these, and to draw a correlation means relating something to something. So all these kind of a parallel between metallic flavors, they will have beany flavors, they will have astringency flavors, while as these nuts, they will have bitter taste, pungent, dry. Similarly, these tubers, they will have kind of these substances. So this is called as a quantitative analysis. We use different kinds of sensory analysis. One of the sensory analysis techniques is QDA, which is called as or RETA. Different sensory tools. So that is not the topic right now. So, but if you want to understand more, link presentation where you can. That's altogether a completely different. So, my is to make they come with different kinds of astringencies, or they come with any flavors, they come with astringent taste, bitter taste, soapy flavors, a lot of into play. So then, first and foremost, what is exactly and what is exactly what is this pungency, what is this stuff and everything else. Astringency is a sensory perception. Mechanosensory perception means which has mechanical aspects in, and it has sense in. Combination of senses and sensorial senses also. Combination of these two. This is a Latin word which means stringer or. So, if nature, if some substance in nature, so that means it will have either oral dryness. Oral dryness means whenever we place it on our tongue, whether we want to taste it, your tongue become dry in nature. That is one of the problems or kind of drawbacks associated with plant proteins. So they have a or a gritty taste plant proteins. Whenever we taste any plant, a sandy texture, gritty texture, and they are not so appealing in terms of the taste and texture also. So second, for example, there are certain kinds of phenolic compounds, for example tannins, for example catechins, which are present in different kinds of coffees, which are different kinds of teas, or unripe fruits also. So I don't know how many of you have tasted a green apple. Go to, if you, or if you go to also the people apples, pluck at ripe stage, you will feel lot of sensations. So it will have obviously high of different substances which will make you love it, but at the same time when you consume that green apple, when you consume unripe, same is true with gum. So you will have this dryness in your mouth, you will have gritty feeling in your mouth. Our, which is the main organ which perceives the taste, and it has different kinds of with a nerve called as nerve. It's our brain, and from the brain means even if there is no taste, still the sensations or still the electrical signals will move from tongue to the brain. Okay, these are different kinds of cell layers, epithelium cells. Oh, the biological aspect. The particular aspect of astringency is that in biological systems, so please be attentive with this one, so because if you understand this, half of the job is done. In biological systems, for example, number one, we take our eyes. Number second, we take our oral cavity. And number third, we take our joints. Please note, this is very, very important to understand. So if our eyes, if consideration our cavity, or if we take into consideration our joints, we see fluids there. In eyes, we see, oh sorry, eyes, tears. In our oral cavity, we see in nature. Why has nature placed all these eyes? Why has nature placed saliva in our mouth? Why has nature placed synovial fluid in our? Okay, so the basic idea is to have lubrication. Basic idea is to have lubrication. So that means 1,000 times, 1,000,000 times you blink your eyes, have a problem, or you that your eyelids and your contact are paining. If they pain, so that means it's a problem, you have to see a doctor. Okay, so if somebody's eyes feel like sandy, if somebody's eyes feel like bitty, so then he has to see a doctor and they advise him certain eye drops. Okay, so there is a lubrication between two, for example, our eyelids and our tongue. So tongue comes into contact with our upper palate. Palate means the upper part of our mouth, inside oral cavity. So whenever we consume anything, we want to eat anything, the food always remains in contact between the lower surface, which is our tongue, and upper surface, which is the palate. It will come into contact with two jaws. Okay, two jaws, when they come into contact with each other, that will have a lot of. So similarly, when the tongue and the upper palate comes into contact with each other, they have a lot of friction. Okay, and same is true with joints also. So if there is a failure in lubrication of joints, that's called as arthritis. So there is a lot of pain associated. Okay, so same thing is true in human beings. I mean, when we talk about oral cavity, so this science is called as oral processing. Astringency, which is feeling of oral dryness, which is a feeling of puckering sensation, or which is a feeling of powder feeling, hypothesized layer of lubrication. So when we consume any substance, consume it chop, just break and put it on your, you will see that with it and it gets dissolved fine. So but on the contrary, when you keep a plant based food on your mouth, say for example a chocolate has been made from plant based proteins, the moment you keep it in your tongue you will feel that it is dry, so that means there is failure of lubrication. Okay. So that failure of lubrication is referred to as astringency. Okay. So compounds, whatever compounds they are, for example they can be plant based proteins, plant based associated phenolic compounds. So they come into contact with this mucosal pellicle. Mucosal pellicle is the when we study, I have kept one of the diagrams there, so we'll explain it one by one. So they bind these salivary, so saliva, different kinds of compositions in the in our slide, proteins in mucin protein, that we also take you through that in next slide. So contains mucin protein, and this mucin protein binds with other associated compounds and there is a breakdown of lubrication. Okay. So this mucin it is present in eyes also, it is present in oral cavity also, it is present, this is one of the major systems. Till now, as of now, the research has been largely focusing on, for example, lot of research has been done on, as I said, that these plant based problems, one for off flavors, has off flavors, soy based. So in processing, filtration process, sonication means ultrasound. So you can at these type of problems. So off flavors in plant based foods has been kind of solved, or we have tried a lot to solve this kind of problems, and there have been lot of innovations, lot of patents have been filed all over the world. Okay. Also the fermentation processes, so that means if we are using different kinds of microbial these cultures and let them grow on different kinds of plant based foods, so the microbial fermentation or different kinds of enzyme treatments, they also have been explored in terms of reduction of the off flavors. Okay. But one of the problems that also comes up, second, first one either tried using membrane filtration processes, or we have tried ultrafiltration process, or for example to remove the off flavors in plant based foods. Second, we have tried microbial fermentation, or we have tried enzymatic methods. Third one that has been also explored in the world is that we can use different kinds of additives. So for example, can be added lot of sugar, lot of other oils, trans fats, they can be added, which have better taste perceptions. So if you are adding more salt, if sugar, if you are adding more oil, if you are adding more trans fats, it gives more appeal to the consumers. Okay. So that is why when we have more products in the market which are laid with sugar, people love it. That's true. Okay. So but this comes with a problem that these are not sustainable at all. So people in the world cannot consume everyday foods laid with high concentration of sugars. They can with salts, they cannot consume amounts of trans fats. They are not, and also they, in terms of the food design different kinds, we cannot, I mean bulk of sugar or bulk of salts or bulk of trans fats cannot be added to the food. Okay. Sorry, just wait. So contrary to this, so that means this is off flavors, vis-a-vis off flavors lot done, but when it comes to property, when astringency property, much research has been done, not has been done till, okay, only few research groups in the world have done. So how this, or how do we understand properties. How do we quantitatively assess the astringency in different kinds of food substances? So largely we do sensory analysis. So previously we have seen a lot of people doing sensory analysis for different kinds of stuff, whether what is the texture of the food substances, what is the taste of food substances, whether this is soft, whether it's blah. Fundamentally correct. There's a lot of wherever human beings are, so there's a lot of feel good to me, right? He might love sugar, he might not love sugar, so I will give 7 out of 10, he might, okay, or whatever scales you are using. Astringency, if you have quantitative astringency, one of the first ways, one of the major ways, or one of the good ways astringency is using tribological data. So tribology as tribology, we measure the oil friction, how much amount happens between two surfaces. Here when we are talking about astringency in mouth only, so we are replicating the friction that happens between two surfaces, tongue and upper palate, okay. So it kind of molded. I will explain this in the coming slide. So then we can also have different kinds of electrochemistry techniques. So as I said that when we taste any kind of food, it's a sensation, it's a mechanosensory perception. It's the electrical sensations that go through trigeminal nerve from our tongue to the brains. So those electrochemistry comes into play where we fundamentally understand the quantitative data of different kinds. For example, we artificial tongue university. So the amount of these ions which are censored, it's a kind of a sensory analysis. Not that well, this is different. So another is sensor analysis. So it's a kind of a sensor. Artificial sensor. I think I have a diagram. Just make you. We are not yet went into the 100%. So if we have to different chains, conductivity salts present in different substances. So they conduct electrical currents, and those conductivities can be measured. One of the way is to measure it through artificial tongue, or there is another technique which is called as QCMD. It measures the adsorption, so how much amount of substances on the surfaces, okay. So then we can also use cell culture techniques, and then we can also use MRI technique, brain image when he kind of, you see that there's a branch of science which is called as food omics. So are now of the opinion that when it's your mood, okay. So if we just bring a pizza in the house right now, most of the people will smile. We are, if we get samosas or we get others, this is not. So the sensations, the person brain can be by using different kinds of techniques. So for example, by doing coding or image drawn from brain, and then using artificial intelligence also, and then understanding that whether there were any astringent substances in this or not, okay. Yeah. I have, just give me some moment, so just understanding. So just for your understanding, this is Cortez crystal dissipation. The full form of this is this. So it's an instrument. So I have all the instruments listed and how they work in the coming slides. I will explain that. Okay, so now we are, if we have all the background in your mind, so plant proteins, astringency, how do we study it? We study it through oral tribology, we study it through different means. So then we will relate rheology and tribology. Most of the people in our institution also, I see like for example Debo, he is also working on plant proteins. he has done a lot of work on rheology. Same is true with Nivetha. She is also working on plant proteins, and she has also done a lot of work on rheology also. So, rheology and tribology are two associated fields. So, we often refer them to as fundamental, okay, in order to understand the textural perception, sensation perceptions of food substances. In particularly, we will discuss about plant proteins. So, again some background to make things level with my audience. So, I do not know how many of you have read any course where you have maybe understood texture, you have had a course or anything texture. Yeah, it might be a part of it. Think of human beings. I mean, when we talk about human beings, whether that be, say for example, in medical sciences, or whether that be in food sciences or any other, wherever human beings come into picture, human beings are very difficult. For example, in medical sciences, so somebody is suffering from, guy is also suffering from headache. This guy takes medicine, it helps him. Another guy takes medicine, it doesn't help him. Okay, because human bodies are complex. Same way, when food perceived beings, they perceive it differently. Okay, so that is why we always take the help of secondary stuff. By I mean by taking instrumental help, rather than just relying on aspects, or rather than just relying on as a subject when they assess substance. We also have kind of in terms of instruments, kinds of instruments for different. Okay, so texture of any food substance, it has meaning. Okay, it is not just limited to taste, it is not just limited to mastication. So sometimes even just a glance on the food, a quick glance on the food only, without even touching also, tells you that whether it will be soft or hard. Okay, so you have two breads, one of the bread is, for example, stale, and you can see it is stale. Another bread is, understand that hard this should, even if you have not touched, you did not even touch the food product, you did not even taste it. Okay, so the touch sensation and then taste perceptions, or even seeing any food substance, so all these, composed of all these, gives you a perception of food texture. Okay, so composition of all these things gives you a perception of texture. So physiological senses, say for example, using your tongue as a sensation, using your hands as a sensation, using your eyes as a sensation, so you can perceive different kinds of senses through these organs, and you can understand whether this food substance, texture means how it exactly has a kind of, whether this is really hard in nature, whether this is soft in nature, whether this is brittle in nature. Okay, so it's a physical characteristic. Okay, so this food texture, it will generally have different kinds of properties. Okay, it can be, I mean, some person can feel texture in terms of hardness, some person can feel texture in terms of its, say for example, the moment you chew paneer, you will feel plastic in nature. It is you chips, wafer, feel that it is brittle in nature, hard in nature. The moment you chance encounter at any foreign body, hard in nature. With food, that it's not just limited to these properties, it also perceives creaminess, it also okay. In the past, we have of food texture with hard, brittleness, crispiness, all those things. Okay, but when somebody consumes yogurt, when somebody consumes ice creams, and we lick that ice cream, we taste this chocolates, you understand that whether they are creamy in nature. When you taste certain gums, or whether when you taste certain kinds of these Okay, so that is another. Or when you consume, for example, or any other plant based substance, you can understand not. Okay, therefore the way here is that efficiency is not just only in the tongue, it is textural. Okay, so that's why we had the we the this sensory perception. The texture also comes into play. Psychological perception. So is a physical perception and a means feel all these properties, various properties. Okay, so they might be, for example, they might be tribological properties and sensory properties combined together. Idea of the textural property of the food substance. Okay, if we now have to study in real life world, that means, as I already told you, that we do not rely on human beings as such. We cannot rely on human beings because when we use humans as tools and the there are certain instruments, for example, analyzers, we can use meters, rehometers, different kinds of food. Okay. So actually texture any food substance measured by using beings. So sensory perceptions can be by doing hedonic. So how many of you know about hedonic tests? Hedonic ratings. Of 1 to 10, we rate different kinds of food substances. Sorry, for students, for example, to make new or a new food product. So then people taste it. Sensory how good is of scale of 1 on 5. Soft, is it as hedonic? Okay. Time intensity, how much time somebody to eat any food, how much substance, how much time it takes for any person to chew any food substance. So that another kind of a sensory analysis, but most commonly we use this hedonic scales. Okay, so this is subjective in nature. Subjective means it is dependent upon the person who does. Okay, can change. If we have the same food, it will have some same characteristics. Okay, they will change. But if I have to food substance decide upon its hardness and another same hardness give code, that means it is subject with the we call them panelists. Sensory analysis, we can have different kinds of panelists. Either they are panelists or do not know anything subject at all. On the contrary, if we do objective testing means we do it through different kinds of instruments. For example, one of the ways is to do texture analysis. We get a TPA curve, that's called texture profile analysis. It tells you how much hard any substance is, how much soft any substance is, how much is there. Okay, a lot of things can be concluded from a texture profile analysis curve. Okay, so then we can understand mechanics. Mechanics means how exactly a solid behaves when different kinds of forces act on it. Some things can be understood and be understood. So our main focus here is to relate rheology and tribology with the texture and oral astringents. The most thing we will be studying here. Okay. Okay. So also one more thing that you have to keep in mind is that the perception of the food substance, if we have to take into account that how human beings perceive food substances, it is not just a matter of one phase, or it's not just a matter of one decision. Okay. So when we consume any food substance, it involves number of phases. Those phases we have to study in terms of tribology, in terms of rheology in our coming slides. But just to take you through here, when we take the first bite of any food substance, we understand, number one, is it hard in nature, is it viscous in nature, or is it brittle in nature? This is the first thing that any human being perceives. Okay, when we take the first bite of any food substance, so you have a chips, when you take the first bite, you understand that this is not so hard, it is brittle, it is not viscous, it is elastic in nature. Things that you can understand on the bite. And all that, what is its shape and what is its size. The food substances can also be understood. So if you are having, say for example, roasted chana, it is a different kind of, it has a different in your mouth. So then second thing that happens after you first bite, you just take it into your mouth, which is referred to as masticatory. Masticatory means the moment your teeth or the moment your tongue and palate come into action. Okay, start completing it, that's referred to as mastication. Mastication is a process of chewing. So those stuff comes into play when we product. Okay, understand whether this product is in nature, whether this product has more chewiness, whether this product has more gumminess. Okay, also this shape and size can also be detected when you are masticating any food product. Okay, following mastication, when the food product is broken down, you understand that whether this food product can be easily broken down. If you are consuming rice which has been cooked immediately, or the rice that has been cooked yesterday night and it has become little bit harder, little bit stale, it will take different times to break it down. So chewing is different. When you for a period of 10 minutes, for a period of 15 minutes, so the chewing rate for both is different. True with meat when you dry it from poultry, true with meat when you dry it from, for example, this sheep or goat. There is a difference in chewing of the two foods. Okay, so the rate of breakdown can be understood just immediately after you start masticating. Okay, so then type of breakdown, how much force you need, this absorption and mouth coating. This is something very important to understand here, is that when you are consuming any food substance, for example a chocolate, and then you are consuming any food substance, okay, more from your mouth coated of the chocolate in your mouth than the coating of rice in your mouth. Okay, so this is generally because of the perceptions that your mouth chocolate melts immediately, rice, the melting process also dictates how much amount of. Okay, so this food texture, now here this food texture, when we are consuming, we have to generally concentrate, all our slides will focus on oral processing. So even if we concentrate on rheology, even if we concentrate on tribology, or if we concentrate on astringency, all these things have to be correlated to oral processing. Oral processing means what happens to food substances in your mouth. That has to be taken into consideration. Whatever slides we are dealing with, even if we are not plant proteins, or even if we are discussing some other, it has to be always correlated in your mind to oral processing. Okay? So this food texture, if we correlate it to and the structure of the food and mechanical properties, biological properties, how much we can understand the oral or saliva, we can understand the time it takes for residence, or understand how exactly the oral processing takes place. For example, for different kinds of food substances, the oral processing happens differently. For chocolates, it happens differently. For wafers, it happens differently. For rice, it happens differently. For flakes, it happens differently. For nuts, it happens differently. Okay, so that all these things can be correlated to food texture versus oral processing stuff. Shall we take a break? Okay, we'll take a break for some time, 10-15 minutes. Okay, so welcome back. We just took a quick break, so I hope you all guys are now recharged. So we discussed how exactly we perceive texture through our different kinds of senses, and when mostly relating it to oral cavity or oral processing. So now we want to also discuss how exactly we need to measure, or how we can measure or sense texture through different kinds of properties. So one of the ways to measure texture is through mechanical approach. So as I said, that the texture is not just a mere feeling, it is not just a mere taste, it's a composed of lot of properties that come into play. And bulk mechanics means whenever we are dealing with fracture of any food substance, that means we have to break down the food substance, the internal structure of the food substance is broken down, and largely we take into account large strain rheology. Large strain rheology means whenever any material fails. So that means we are using a larger strain. So I think I will have to repeat it number of times. So whenever any food substance, or any substance for that matter, not just food substance, when it is under any force, force per unit area is referred to as stress. The amount of force that is applied on any unit area is referred to as stress, and the resultant change, that happens in its length, that happens in its volume, is referred to as strain. Okay, so the stress and strain are related. So that means whenever we apply any force on any food substance, it results in any deformation. So that deformation is studied in terms of this resultant strain. Okay, so then there is another term which is referred to as shear stress and shear strain. So we will go through that. So here, so if any food substance is, if we apply large amounts of shear on any food substance, so that means we are applying lot of strain, is resultant strain is very high. It results in the breakdown of the food substances. अगर आपने elasticity का concept पढ़ा होगा, I mean in your primary classes also, we see that for example when any food substance, when we apply large amounts of force, suppose if you have a pen and you take what do we call, which is refill, if you take a refill out of it, if you keep on bending it, it will kind of come back to its original state. But when you apply lot of force on it and it exceeds certain limits, it breaks down. Okay, so a bulk mechanics always deals with whenever we are applying high amounts of stress on any food substance, whenever we are applying lot of force on any food substances. Okay, and then we can also study the texture through rheology. It's a very specialized field. Rheology studies the deformation of any food substances. It studies the flow properties of any food substance. Rheho means to flow. So technically, rheho means to flow and logy means to study. If we study the flow properties of any food substances, that is referred to as rheology of the foods. It's also a mechanical engineering concept, largely studied in mechanical engineering or material sciences. And then foods, when I was in my masters, we had a concept of food material sciences. So we always think of foods as materials. So if you think of foods as materials, these all picture comes into play. Texture properties, rheological properties, tribological properties, other mechanical properties come into play. Okay. So then psychophysics. Psychophysics, as I told you in the previous slides, that the texture is not just a perception of mechanical properties, it is not only a perception of sensory properties, it also is a perception through your psychology also. Okay. So those psychophysics concepts can also be studied in order to understand how the food substances are, what is the texture of the food substances, that can be studied. So then there is a new concept called as psychorheology. So the rheological properties of any food substances are correlated with the sensory perception of the food substances. If we combine them together, the instrumental data is coupled and discussed in terms of the sensory perceptions, in terms of the psychological perceptions, we call that as field as psychorheology. Okay. And then obviously the oral tribology, which is our main focus. So here, from here you can kind of draw two outlines. We will be discussing more about food rheology and we will be discussing more about oral tribology. Okay. So food rheology means flow properties, food rheology means flow behavior of any food substances, and tribology means frictional properties of any food substances. Okay. So lubrication properties of any food substances, which is the talk of the town. Okay. So why do we want to study food texture? Is it really necessary? So my funda in life is also that if something is not necessary, we should not do it. Okay. So how exactly food texture is important to alternative proteins? Is it really important that food texture has to be studied in the context of alternative proteins? Yes. Number one, why? As I said, that food proteins are not consumed only as powders. In the world, not just limited to India, in developed countries or in certain developing countries also, food proteins are not just consumed by consumers as powders. Okay. For example, some athletes, some bodybuilders, they consume whey proteins as powdery substances. But if you have heard of different companies, so for example, one of the companies is Beyond Meat. How many of you have heard of Beyond Meat? It's a US based company. So they are making plant based nuggets, plant based burgers, plant based ham, lot of substances are there. So if we are just moving away from powdery substances to real foods, okay, so plant based burgers for example, is a chicken nuggets, so they need to have a texture which will have better cheviness, which will have better juiciness, which will have better bite properties. Okay. So plant proteins are dry in nature, plant proteins are astringent in nature, anything make out of plant proteins will be dry. That is also a problem, as when in the starting, that when we are, they have water holding capacity. Okay. On the contrary, animal proteins, if we talk about animal proteins, they have high water holding capacity. Surface hydrophobicity in case of plant proteins is very, very high. We have to limit surface kind of chicken nuggets, alternatives to chicken nuggets. They are very dry in nature, they are brittle in nature, they are not soft and juicy in nature. Okay. So therefore we have to take into account the consumer preferences. The consumer preference is, number one, the texture should be such that it should be juicy in nature, it should be better, it should have better cheviness, and it should have a better bite. Okay. So second, alternative proteins, whenever we discuss about alternative proteins, alternative protein based meat, alternative protein based chicken meat, or alternative protein based fish replicants, everything else, they have to mimic the sensorial properties of meat. Okay. So these things can only be achieved. So whatever we discuss, this textural properties, cheviness, juiciness, blah blah blah, and then it has to mimic properties, it has to meat, that can only come between textural properties of our plant based substances and real world substances. Real world in the sense, I mean, for example, meat. Okay. If we have to kind of draw parallels, plant based meat, for one of the is that proteins structurally are different than animal based proteins. We have to keep that in mind. So, plant proteins are typically globular in nature. So, plant proteins are typically globular in nature, okay, while as animal based proteins are, they are fibrous in nature, okay. So, they have fibrillar structure. So, for example, let us put it this way, they are along length scales, okay. So, plant is not length scale, they are all globular in nature. Kind of understand in your mind that in nature, while as rest of the proteins which have to mimic, which they have to mimic are along length scales, okay. So, length scales help them to have a different kind of structure, while as globular stuff helps them to have a different kind of structure. So, now if you have to, if meat proteins have acid proteins, so they are, one is north pole, another is south pole. So, the globular proteins have to be changed into fibrillar structures, okay. So, that is why we need, I mean you can read it again when we talk, I mean it is a very emerging technique. So, probably when you go back home, so one of the key takeaways from here is when you go back home you can study about meat analogs. When we were working in Shanghai, we were changing globular proteins into fibrous structures and changing isotropic structures into behavior. So, that this thing you can study when you are going back. So, that maybe this is kind of a research area that you can explore, okay. So, whenever we kind of use plant proteins in industries, so we need to have various modifications either through shear cells. So, there as of now two techniques are in the globe, the world. Two, for example, let us say that if we talk about meat analogs, so two different techniques are used around the globe. One is high moisture extrusion, another is shear cell technique. So, these two techniques are modification techniques and high modification techniques. They require a lot of, and those substances can be changed. So, that means these globular structures can be changed into structures by using shear cell techniques. Okay. So, how this is done? Number one, apply high amounts of heat, used as a modification process, and then pressure is used to reorganize of proteins is changed into structure of the protein. So, one of our PhD students is also exploring different means of developing from bean proteins, fibrils we want to use for different kinds of, for example, in pickering emulsion. Second, so whenever we want to make any new market, if we have to make a new product for the people, it has to be optimized, it has to be an optimized product. So, I do not know how many of you have probably ever in your BTech, in your bachelor's, done a simple project of making, for example, say cookies from millets. Suppose just an example that we want to make cookies from millets. If we want to make millet, if we want to use millet flour, generally traditionally we are using wheat based flour to make cookies, okay. So, now if we are replacing millet, please be attentive. So, if we are replacing wheat flour with millet flour, so we will use, for example, 1%, then we will use 5%, 10%, and see that whether it is possible to make cookies or two are being made, whether they are color is good or bad. Let us assume that amongst 1%, 2%, 5%, 10%, 5% was okay. So, this means we have optimized it in terms of taste, in terms of texture, in terms of color. We got, for example, 5%, up to 5% substitution is okay. That means if 10 grams is 5% with millet flour, that's called optimization, okay. So same way optimization has to be done in terms of rheological properties, in terms of tribological properties, in terms of textural properties. When we cannot as of now 100% replacement, if 100% replacement of plant proteins is done, so we are still not able to achieve better rheology, we are not still able to achieve better tribological properties, we are not still able to achieve better textural profile analysis of the products. Okay? So here I want to emphasize is that we have to optimize it. So example, one of the research that we did in our lab at University of Leeds, which I will show you the paper also, is partially replacing or adding different kinds of plant proteins to smoothies. If we are making a smoothie, we are adding protein, we are adding whey protein in it. Plant protein, animal protein. How much amount of pea protein, up to what level of plant proteins we can add, up to what level of soy proteins we can add, up to what levels of whey proteins we can add. Okay. So then, for example, let us say that we were able to achieve 2.5%, up to 2.5% we can add pea proteins in smoothies, up to say for example 3% we were able to add soy proteins, up to 10% we can add whey proteins without compromising seriously on taste, without compromising seriously on texture, without seriously compromising on tribology. So that means we have to kind of do estimations, we have to do measurements, and we have to optimize how much amount of replacements we can do. Okay? And then ultimate goal. What is the ultimate goal? So when we are relating texture to all proteins, is that when we are using alternative proteins, giving consumers alternative, so to achieve the same textural properties, they have to achieve taste, or they have to achieve the same color characteristics as the replacements are. If I am taking chicken right now, if some of our student comes with a new enterprise startup that I want to have plant based nuggets, they should be able to at least replace it and help me get the same taste, same color, same perceptions. Okay? So that is the ultimate goal. So how that can be done? Number one, we have to have exactly meat like mechanical strength. So if it is like, for example, one of the problems associated with making plant based fish stuff is that is not at all able to withstand deep frying, that is not at all able to withstand cooking, the structure collapses. Still, for example, if we are trying to make chicken nuggets, plant based chicken nuggets, they still are able to withstand deep frying, they are still able to withstand extent of boiling. Okay? So that thing has to, ultimate goal is that we have to achieve those mechanical strength. Juiciness has to be, the chewiness has to be perfect, and it has to be sustainable in nature. So also one more disadvantage with using plant based proteins is that when we are trying to use them in real life scenarios, real foods do not come only with 100% of proteins. Okay? Even if we consume fish, which has very high concentration of proteins, even if we consume chicken, which has very high concentration of proteins, if we consume meat, mutton, if we consume beef, which is having very high concentration of proteins, they come with some added nutrients. So for example, they come with fats, they have very low concentration of carbohydrates or whatever. So here we are trying to replace oils also. When we are trying to make chicken based nuggets, or when we are doing high moisture extrusion, we are trying to reduce oil concentration or adding oil in it. Sunflower oil can be added or any other oil can be added, but it is not able to replicate the real world fat which is present in meat. So because that the proteins in meat are not only responsible for the textural properties, the proteins in meat are not only responsible for chewiness of the properties. The fat which is marbled meat means which has got protein layer, fat layer, that is called marbling of meat. So if the white layer is there in between two muscles, that also helps you to have a better chewing property. So that still is not, we are not able to completely 100% mimic those things. Okay? And sustainable proteins means we have to use plant based substances which are, say for example, if we want to isolate proteins from rice, probably that is not a sustainable way to do it. To start with, it is okay. To start with, to do research, fundamental research is very important. Okay? So we can do research on rice also, we can do research on anything, but then it has to be sustainable also. Sustainable is, for example, leaf based proteins. Sustainable is, for example, millet based proteins. Sustainable is, for example, pea based proteins. Okay, they are sustainable in nature, or we can use fungal this natures, or we can use cell based natures. And then all these things combined together, consumer key consumer drivers, structural properties, optimization properties, goals, they all have to take into account the consumer acceptance. If its consumer acceptance is very high, our goal is done. Okay. So these food texture and alternative protein relevance, the relationship between food texture and alternative proteins, whether they are relevant to each other or not, this dictates whether we can be able to replicate, whether we can be able to innovate. One, replicate means whether we can be able to do it on whether we will be repeatedly or not, whether we will be able to do more innovations in it. So this will be one of the key successes for achieving this will be one of the drivers for achieving success of plant based substances. Okay. So now coming to food rheology. So it's a very vast lecture today. So you have seen number of products in the market. So for example, we have seen molten chocolate, we have seen, for example, tomato pastes, we have seen ice creams, we have seen mayonnaise, we have seen juices. All substances behave differently. All substances behave differently. When it comes to stirring, for example, very easy to kind of imagine. When we stir milk, it is different. When we stir honey, it is different. When we stir mayonnaise, it is different. When we stir tomato paste, it is different. Okay. So how exactly food substances or any other materials behave when they are deformed, or when any forces act upon them? That branch of science is referred to as food rheology. It can be either for liquid foods, it can be either for solid foods. Either it is for liquid foods, either it is for solid foods. Okay. Solids also deform under stresses. Now it depends upon what kind of solid we are imagining. If it is iron, there will be no deformation at all, almost negligible deformation, till extremely high amounts of forces or high amounts of stresses are taken into account. Okay. So generally when we consume these food substances, whenever we consume these food substances, we generally do not kind of take into account food rheology at all. Okay. We consume different kinds of foods and generally forget about rheological properties. So if the tomato paste or tomato puree, for example, when it is in a glass bottle, if it is too liquid in nature, if it has got very high if it is viscous in nature, you will feel like it comes out as a water. Okay. So that people will not prefer it. So there is a particular consistency which people like. So the moment you try to take it out from the bottle, you have to apply certain amount of force, it comes out and just spreads. Okay. So that is a particular amount of behavior that different kinds of food substances have. So any food substance, when any external force is applied on it, it will deform. So that study is referred to as rheology. Okay. So if we take into account any food substance, and the force acting on it can be applied differently. It can be applied at 90 degrees to surfaces, okay, or it can be applied parallel to the surfaces, okay, or it can be applied in different directions. Say for example, we can apply it in circular motions or oscillations. Okay. So largely this one is mostly important. So if you want to know more about it, probably you can go back to study this also, but this one is more important when it is applied tangentially or not this at 90 degrees at an angle. This force is referred to as shear stress. Any force for that matter, force per unit area is referred to as stress, and the force which is applied tangentially, which is applied at different angles, is referred to as shear stress. The resultant strain, resultant deformation is referred to as shear strain. Okay. So whenever any force is applied on any material, it deforms and it begins to either flow, or it either loses its shape, its volume changes, its area changes. So either this shear strain is calculated in terms of del upon or L, or it's in del V upon V, or del A upon A. That means original length, change in length, original length, change in volume, original volume, change in area, original area. So this happens, these things happen only when external forces are applied on it. Okay. And then one more advanced way is to apply. So I mean, this is not always true that the forces will apply perpendicular to each others or at different angles. Sometimes the forces also apply in circular motions, which is true with stirring, which is true with mixing. Okay. So that shear stress and shear strain translates to oscillatory stress and oscillatory strain. Oscillatory stress and oscillatory strain. So we can study rheology in terms of shear stress and shear strain also, and we can study rheology in terms of oscillatory stress and oscillatory strain as well. Okay. So done. This I will not take much of time, because I think most of the people have an idea of different kinds of fluids. So I will just have a quick glance of it. This is just a background in order to understand rheological concepts. Okay. So but I mean, my advice is that if you have to read more, or if you have to understand rheology more, we should have an understanding of all the flow behaviors of any fluid substances. Just I will be very quick here. So the fluids, please note, fluid does not mean liquid. Please note it. Fluid does not always mean a liquid. Fluids can be solids also. Solid, by the way, does not mean iron only. Solid means yogurt also. Solid means cream also. Okay. So liquid means obviously milk, water, or juices, or substances like that, which are both included in fluids. So these can be either Newtonian in nature, they can be non-Newtonian in nature. So in Newtonian fluids, what happens is that the shear stress and shear strain are always have a straight line relationships. Okay. That means in spite of how much amount of stress you apply, strain will always be constant. Okay. Whereas that's not true with non-Newtonian fluids. So that means they are typically of different types. So they can be, for example, time dependent fluids, time independent fluids. They can be thixotropic, they can be dilatant, they can be thixotropic, dilatant, rheopactic. So you please just read this. There is a relationship for different kinds of fluids, shear stress versus shear strain. Then you will have this as non-Newtonian, this is a Newtonian fluid, and then we will have different kinds of curves. Okay. So in order to understand rheology more, you should have an understanding of this one. So but if we kind of go back to everything on this, probably it will take a lot of time to make you understand. So please, at least once you just understand this models. Okay. So coming to rheology back again. So how do we measure rheology? We usually measure rheology by using a rheometer. Okay. So then there are different kinds of rheometers, which quickly I will take you through. So if you happen to visit in our labs, maybe today, tomorrow, or day after tomorrow, you will see a rheometer there in our labs, and maybe you will operate it also, and you will see by your if you operate it, you will see that there are different parts of a rheometer. Okay. So some of the more important things about, I mean, although everything in this rheometer is very important, so important things, most important things in this rheometer are the measuring systems, which are also referred to as geometries. Okay. So the lower plate, this one, the lower plate here, which is stationary, it does not move. Okay. On this stationary plate, we keep our sample, we spread our sample, and then we use different kinds of measuring systems, either a cone and plate geometry, either a parallel plate geometry, either a cylinder geometry. So this depends upon the type of the food substance that you are using. If you have a liquid substance, you can use a cylinder. If you have any other substance, you can use cone and plate, you can use a parallel plate. And for general experiments, which are, say for example, plant proteins, which we generally use powdered substances as such, or starch based substances, we can use parallel plate geometry, or we can use cone and plate geometries also. It depends upon different kinds of experiments that you are doing. Okay. So, this cone and plate geometry, this parallel plate geometry, and this cylindrical geometries, they are changeable. I mean, you can just take out this portion. This is the geometry. Probably this one is a parallel plate geometry if I am able to see it properly. So, you can remove it, fix another geometry, take it out, fix another geometry, so that you can interchangeably use depending upon your own experimentations. Okay. And then this rheometer, it does not just only change the shear stress, shear strain values, it does not change only oscillatory stress, oscillatory strain values, we can also incorporate temperature in it. So, that means the temperature can also be changed or varied. What happens to the food substances real times, if we want to change the temperature of the food substances also, we can have temperature controlled rheometers also. So, depending upon what type of experiments you are using or what type of equipment you have, we call them as stress controlled rheometers, temperature controlled rheometers. Okay. So, this one of the examples of this one is strain controlled rheometer. One of the examples that I purposefully kept here is that this is a strain controlled rheometer. So, in this one, in this instrument, we can vary the speed or we can vary the angles at which the force will act on it. Okay. So, then we have different kinds of sensors in the rheometer which sense the different kinds of properties. That also we will take care of in the coming slides, that when we are studying rheology through these equipments, what is the output, how do we actually sense rheology of any different food products. Okay. So, as is true with strain controlled rheometer, we also have stress controlled rheometers. So, that means we largely focus on stress instead of strain. Okay. So, different kinds of forces can be applied and different kinds of stresses can be applied, varying stresses can be applied on the food substances, and then we can understand what happens to the food substances when different kinds of stresses are used. So, either a pressure controlled rheometer can be used, or a rheometer with UV radiations can be used, rheometer with humidity values, changing humidity values can also be used, or a rheometer with temperature control can also be used. Okay. So, sometimes what happens is that when we are dealing with emulsions, for example, so there any minor change in temperature will have devastating effects on rheology of the food products. Okay. So, in order to have minimal effect of temperatures, we try to control temperatures. I am not sure whether you have heard of it. This is a general phenomenon, when we are stirring any food products, more stirring, more friction, more heat generation. Okay. So, same is true with this rheometers also. Whenever we are using any rheometer, when we are doing any experiments on rheometer, it results generally in temperature fluctuations also, even if those fluctuations might not be very high, but there will be fluctuations in temperature also. Sometimes in our experiments, we just want to negate all those temperature effects, so we can use temperature controlled rheometers also. So, as I said, okay, when we are doing experiments on rheology, or when we are doing experiments on rheometer, the resultant values that we get is in terms of G dash and G double dash. Okay. G double dash gives you how much substance is elastic in nature, and G double dash gives you how much amount of viscosity or viscous nature any food substance has. Please note that materials can be either elastic in nature or materials can be viscous in nature. So, that means when we are applying any force, when we are applying any stresses on the food substances, they regain their shape back. That is called as elastic nature. So, when we are applying any force, or even if we are not applying any force on it, water for example, it is highly viscous in nature. Okay. So, food substances generally lie in between elastic and viscous nature. So, therefore, we refer to them as viscoelastic natures. Majority of the food substances have viscoelastic natures. Okay. So, the resultant or the data from the rheometers is generally drawn in terms of values which are referred to as G double dash and G double prime, loss modulus and storage modulus, which is elastic nature and viscous natures. Okay. Now, then there is one more value which is referred to as tan delta, which is the ratio of G double dash by G dash. Okay. So, this tells you, the ratio tells you whether any food substance is highly viscous in nature or any food substance is highly elastic in nature. The ratio of the two tells you whether any food substance is highly viscous in nature and whether any substance is highly elastic in nature. Okay. So, now if any substance is elastic in nature, if it is 100% elastic in nature, you see, ideally elastic will be that G asterisk or G double dash. If G double dash and G dash are equal, they are completely equal. So, that means this is elastic in nature. Okay. If G dash is equal to 90%, 90, this is known as phase angle. Okay. That is why there is degree there. It is known as phase angle. So, it will have a perfectly viscous nature, but since food materials lie in between elastic natures and viscous natures, their values generally lie between 0 to 90 degrees. Okay. If it is more towards, for example, 90s, so we will have it as a fluid like properties. If it is more towards 0 degrees, we will have a solid like or gel like structures. So, this is what you have to remember in rheology. So, that is here. If the phase angle is equal to 0, it will have perfectly elastic nature. If the phase angle lies between 90 to 0, but more off towards 90, so it will have a viscoelastic nature. If it is more towards 90 degrees, so that means it will have 100% viscous nature. Okay. So, as I said that instead of just applying forces parallel to each other, instead of applying forces at different angles, we can also have forces which are oscillatory in nature. Okay. So, that tests if we are applying forces which are oscillatory in nature, that means we are applying frequencies means rate of change of RPMs, for example, rate of change of cycles, for example. Okay. So, those tests are referred to as amplitude sweeps or frequency sweeps. Okay. So, amplitude sweeps or frequency sweeps, they generally tell us the deformation behaviors whenever we are using oscillatory measurements in rheometers. Okay. So, this is generally done in order to determine the yield point, also the flow point. So, when any food substance undergoes deformation, so it will have, for example, say it will sustain for a period of time and then it will break down. So, this is known as yield point, and then completely 100% it will flow like a liquid, that is known as flow point. So, this I will explain in coming slide, just wait for me. Okay. So, frequency sweeps, they are time dependent. So, that means we have to take into account time also. The more you keep on oscillating it, for example, dahi, yogurt, the more you keep on oscillating, the more you keep on applying oscillatory stresses, it turns more into viscous natures. So, those types of substances are referred to as time dependent fluids. Okay. So, time dependent behaviors and non-destructive in nature. So, they are calculated by using frequency sweep tests. Okay. Here, to understand more about storage modulus and loss modulus, that means if we have to understand more about G prime and G double prime, please see it here. Now, first, flow curve of viscoelastic material, note the differences, it is same, flow curve of viscoelastic material. So, that means as of now we are taking any food material in concentration which has properties both viscosity and elasticity. In both the cases, in A case also, in case B also, we are taking into account a food material which has properties of both viscosity and elasticity. It might have more viscous nature, okay, it might have more elastic nature, okay, so that we will understand. Now, one of the regions in rheological data is referred to as LVER region, that is known as the stress and strain, G dash and G double dash, they are almost same, they run parallel to each other. Okay. In case of a viscoelastic behavior, in case of viscoelastic properties, where elastic nature is more, So initially in LVR region means where stress and strain are applied such that there is no deformation. This means low viscoelastic region. Shear stress and shear strain are not very high. Okay. Beyond LVER region, when shear stress and shear strain increase, beyond certain regions, you will see that there is breakdown. That breakdown is referred to as either yield point, or it is referred to as yield stress, or it is referred to as linearity limit. So that means stress and strain are linear to each other, but the moment we increase stress, strain also increases. There is completely breakdown of the regions. In this one, G prime is more than G double prime. G prime is more than G double prime. So that means the elastic nature is more. When we are applying forces, initially G dash and G double dash run parallel to each other, but G dash is more than G double dash. So this is an elastic nature. On the contrary, if it is a viscous nature, G double dash will be more than G dash, G double dash will be more than G dash. So always in LVR region also, and beyond LVR regions also, beyond LVR regions also, viscous nature will always dominate. Here viscous nature will always dominate, but you see here elastic nature and viscous nature come closer to each other, because this after fracture, when you are applying very high forces, elastic nature completely breaks down and it behaves like a viscous fluid. You see here, this is the actual point that I wanted to make you understand here. Because the food substance is already viscous in nature, the viscosity profiles or viscosity performance outnumbers or outsmarts this G dash elastic behaviors. Here beyond certain regions the elasticity is lost, and this elasticity turns more towards viscous behavior. So therefore G dash values and G double dash values coincide together. So in plant proteins also, if for example your G dash values and G double dash values are like this, after a certain period of time G dash values will be combining together and they will lose plant proteins upon high mixing with water, with other substances, with smoothies. They will try to be viscous in nature rather than being elastic in nature. Okay. So this is again the same thing. So if you are having small LVR regions, so that means shear stress and shear strain are too low. But if you are having larger LVER regions, so we can see that the elastic components can withstand for a pretty long period of time. This one, elastic components can remain for a pretty long period of time, but while as this components, they don't remain for a pretty long period of time. They behave as viscous natures upon larger LVR regions. So when linear viscoelastic regions are completely predominantly of viscous nature rather than elastic natures. Same things, substance can be viscoelastic solids, either the substance can be gel like or a substance can be viscoelastic liquids. Okay. So in case of this solids, the storage modulus, that means the elastic components are high, loss modulus increases, so that after a certain period of time the this viscous natures are very high. This is the relationship between the two. Phase angle means relationship between G dash and G double dash. Whereas in case of gel like structures, they are almost in between the viscoelastic range. They are neither 100% elastic, they are neither 100% viscous. So therefore G dash and G double dash are always same. Okay, they don't change. Whereas in case of substances which are liquids in nature, the viscous natures will be always very high than elastic natures. Okay. So coming back to Now, what importance this rheology holds from farm to fork? Number one, rheology can be used for product development and texture designs. So, if we know better rheology, if we know better textures, if we know better viscosities, if we know better mouth feels, we can have better product developments. Product developments means if we want to make a new kind of product in the market. So, also these proteins, whenever we talk about plant proteins as such, plant proteins have to make gel like structures. So, the gelation is an important phenomenon associated with plant proteins. So, some of the animal proteins make better gels than plant proteins. So, therefore, we modify plant proteins through shear extrusion, through other heat treatments, so that they can also make better gels. So, but we need to have control over how much amount of gel formation should happen. Okay, with heating, how much they should form the gels, how much thickening should happen, how much emulsification should happen. So, therefore, when we have to talk about plant proteins and bring them into market, we study different characteristics. One is, for example, gelation of proteins, we study, for example, emulsification properties, we study other properties also. Not what is that happen? Did I do something wrong? Okay, so different kinds of properties are studied. Okay, so then, as I said, in order to make newer products, optimization of the quantities, optimization of different behaviors or amount that has to be added in extrusion during mixing or 3D printing, that has to be taken care of. Okay, so if we optimize the properties, it also helps us to achieve, for example, how much amount of fibrous structure we will be able to achieve, how much amount of moisture it can retain, how much amount of temperature we need to change, different kinds of properties. So, that can be achieved when we know about food rheology. So, probably I think this quality and assessment we can, I mean, this has no direct influence, but generally when we have to use different kinds of plant based substances have got lesser stabilities over animal based substances. If you incorporate animal based substances in emulsions, they will have a better storage stability, but plant based substances, when they are used as emulsifiers, they have got lesser stability. So, when we are doing long distance transportations, whether they will be really able to sustain for a long period of time or not. So, we can predict the cooking properties, the transportation properties, we can predict whether they will sediment or not, whether they will undergo syneresis or not. Syneresis means, yes. So, for example, if we have starch based substances, whenever we keep them under low temperatures, they release water, the water comes out, that is known as syneresis, and particularly in starch it is known as retrogradation. But if we talk about plant proteins as such, so when plant proteins undergo different kinds of processes and we add water to it in order to make them more juicy or any other product that we made up, what happened, if we have to transport them for long distance markets, whether they can really remain as such, whether they can maintain that structural integrity or they will release amounts of water. So, that is called as syneresis, that can also be studied. Okay. And then finally, these sensory and consumer acceptances, that this rheological properties, they correlate to creaminess, they correlate to chewiness, they correlate to spreadability, and that has to be also taken care of. Okay. Now, we come to more oral processing, and then I think we will move slowly to tribology also. You see, this graph in particular. So, thickness of any product, so whether any food product is thick or whether any product is thin, it can be predicted through low viscosity or high viscosity. So, if any product has got low viscosity, so that means it is not thick in nature. If any product has high viscosity, that means it is thick in nature. Okay. So, that also determines how much amount of mouth feel you have for any product. Okay. So, juices, they have a very less mouth feel because they are having more viscous behaviors, they do not remain in your mouth for a long period of time. Contrary to that, cream based products, yogurt or any other ice creams, they remain in your mouth for a longer period of time. So, they have got higher thicknesses. Okay. So, similarly, stickiness and mouth coating, they can be, they also come up with viscosity profiles. So, that they whenever we consume any food substance, whenever we consume any food substance, it comes into contact with saliva. Whenever we consume any food product, it comes into contact with saliva and it gets coated, or bolus formation happens. So, this coating of food materials by saliva or formation of the bolus after we chew it, that stickiness, the mouth coating, is predicted in terms of extensional viscosity. Okay. So, this we will read in the coming slides. So, also as I already told you, if a food substance has very high sweetness, if it has got very high flavors, they are positively correlated. I mean, people love those types of foods. If we have very high sweetness, generally people love to eat, eat these products, and if some products have very high flavor perceptions, they also have very high appeal amongst people. So, because of higher viscosity, I mean, when we add sugar to any food substance, when we add other types of sweeteners to any food products, when we add honey to any food products, the viscosity increases. So, because of higher viscosity, the sensory perception or the residence time in your mouth is higher. So, therefore, the perceived sweetness is high. Okay. So, therefore, please note, the sweetness and the thinning behaviors or shear thinning. So, that means when we are putting it in our mouth, we are masticating it, we are coating it with saliva, the sweetness and shear thinning are inversely proportional, more sweetness, less thinning. Okay. So, here you see the viscosity profile, viscosity drops when we are applying more shear rate. So, that means in our mouth when we are consuming anything, the more amount of mastication we do, the more amount of chewing we do, the more amount of residence time and more amount of contact with saliva, it reduces the viscosity of any food product. Okay. Please note now here. So, if we take for example jam into consideration and you take a spoon of jam in your mouth, if you take a spoon of jam in your mouth, initially the shear rate is very low. So, that means we are not applying so much of force in our mouth, we are just putting it on our tongue. It will be hard in nature, it will be thick in nature when saliva does not come into contact with the jam. Okay. So, the shear rate is very low. So, at very low shear rates, this jam behaves as a soft gel. Okay. So, we can feel in our mouth that it is thick in nature and it is hard in nature. Okay. So, these generally, if we have to do these kind of testings, whether on a texture analyzer or whether on a rheometer, they are generally done at ambient temperatures of 20 degrees centigrade, which almost mimics the temperature of our buccal of our oral cavity. Okay. So, then as soon as this jam comes into contact with saliva and also the temperature rises, initially when we just put a spoon of jam in our mouth, it is suppose at a temperature of 10 degree, 15 degree or so. The moment it comes into contact with saliva, its temperature rises and shear also increases because of our tongue or because of our teeth. It changes from hardness stuff or thickness stuff to smoothiness to melting to coating. Okay. It gets coated with saliva also, therefore, it feels like smooth in nature. So, the food that we consumed, jam we consumed, it undergoes melting. As soon as it comes into contact with saliva, it becomes smooth in nature, and because of mastication also, it becomes very smooth in nature. So, this phenomenon, under these conditions, you see that it will spread in your mouth. Under these conditions, it will spread in your mouth. So, now if we replicate same conditions and we use a parallel plate in geometry in case of rheology. So, we put our jam here and then initially it is solid in nature, it will have harder texture, it will be thick in nature. The moment we apply stress on it, the moment we increase temperature in it, it will start flowing, it will start behaving as a viscous fluid, and that is what also happens in our mouth immediately before swallowing. So, that means just before our swallowing, it almost completely becomes a liquid like substance. Same is true with chocolate also. The moment we take chocolates, it is 100% solid in nature, it is hard in nature, it is thick in nature, it gets coated with saliva, it starts to deform, it starts to become liquid in nature, it becomes smooth in nature, you love to taste it, and then the moment you start swallowing it, it is almost in the shape of a liquid. Okay, this is how exactly it happens during oral processing. So again, now we come on to tribology. That was about rheology. Now we come about on towards tribology. So in rheology, we study about the flow behaviors. What happens to flow behavior? But in tribology, we study the friction. If there are two surfaces, this is one surface, this is another surface, and in between these two surfaces we have certain material, and if these two surfaces are in relative motion, or one surface is stationary, another is in relative motion, what happens to the frictional properties? That properties, if we study, they are referred to as tribological properties. So tribology is again the way rheology, rheho means flow, logy means study, tribology means rubbing, and logy means study. So tribology is the study of lubrication properties or frictional properties. Whenever two surfaces are in contact with each other, and there are always chances of wear and tear, or there are always chances of high friction. So the study of all these associated phenomena, all these characteristics is referred to as tribological properties. Okay? So this tribology is derived mostly from mechanical engineering sciences. So whenever they make different kinds of materials, whenever they make different kinds of equipments, so when equipments run, for example, these pulleys run, these bearings run, and what happens to the surfaces? So that study is in mechanical sciences. Tribology is also studied, but in case of food sciences, the tribology largely is studied within the surfaces of the tongue and palate and teeth. So that means whenever we put our food substance on our tongue, the food substance remains in between our tongue and the upper palate, and they come into contact with each other. So there is always relative motion between tongue, food, and upper palate. So there is a lot of friction in between the two, and the food in between our teeth. So whatever happens inside our mouth is frictional properties only, those properties are studied through tribological properties. Okay, so these properties give you an idea of whether the substance is creamy in nature, whether there is smooth in nature, whether it is slippery in nature, whether there is astringent in nature. Largely we focus here on astringent behaviors. If we have to study the astringent behaviors of any food substance, we largely do it through tribology. Astringent behaviors cannot be studied through rheology. Creaminess, smoothiness, slipperiness can be kind of an idea we can draw by using rheological data, but astringent data astringency field or astringency outcomes cannot be taken from rheological data. They always have to be taken from tribological data. Okay. Now please note, if the width between two surfaces, is low, please note it. If the width between two surfaces is low and the width between two surfaces is very high, understand? Either the width between two surfaces is very low, tribology comes into play. If the width is too high, tribology does not come into play because there is no friction between the two surfaces, right? So if the width goes very high, this falls under rheological zones. Okay, if our mouth is open, it will have the characteristics of rheology. If our mouth is closed and tongue and food is in between, the food is in between tongue and palate, we will always have the tribological characteristics. Okay. Whenever we are eating food, and we are doing oral processing, it is a composed of oral processing is a composed of tribological properties and rheological properties, because we do not always keep our mouth closed. Whenever we are chewing any food, whenever we are dissolving any food, where we are swallowing our food, we close our mouth, we open our mouth. There are a lot of things happening together. So it's a composed of oral processing is a composed of tribology also and rheology also. One thing. Second, whenever food which is in between our tongue and upper palate, so when it is coated with saliva, that we will study in here also. So whenever we study this I will explain in the next slide. This was lubrication regimens. Okay, so when the food comes into contact with saliva initially, whenever food comes into contact with saliva, it is still in between two surfaces. Okay, so when food is completely coated with saliva, when food is 100% coated with saliva, so it is not in between two surfaces. Food is in between. Food comes into contact with lot of saliva, then that saliva is in contact with different kinds of surfaces. That also has to be taken care of. Okay, so therefore, this lubrication, if the solution is limited, if the saliva is limited, and food is in contact with the tongue and upper palate only, so we have 100% tribology zone. Okay, or the gap width is not too less, too high, so we have a composed behavior. That means it will have some properties of tribology also, it will have some properties of rheology also. So this happens during mastication process. Initially, the moment you put food on your tongue, it is 100% this tribology. When you completely swallow it, beyond swallowing, it is 100% rheology. In between, whatever happens is a composed of rheology also, is composed of tribology also. Okay? So how do we study tribology? So as I said, tribology is about friction, it's about wear, and it's about lubrication. Okay, so the tribology can give us an ideas of frictional properties, it can give us an idea of wear and tear, it can give us an idea of lubrication also. Okay, so this tribology or friction, it is how much amount of force is whenever we are moving any substance on any other surface, how much amount of opposing force is there, that's called as friction. The opposing force to any movement is referred to as friction. So whenever we are swallowing our food, we are taking it backwards, is there any friction on the surfaces? That can be understood by using different kinds of tribological data. Okay, so here whenever we are using this, first I will explain this one, then we will go on to second one. This is more important. So we have a fruit smoothie and we want to incorporate pea proteins in it. We want to make it nutritionally better. Okay, or in order to combat different types of diseases, so somebody is suffering from, say for example, fatty liver, and he doesn't have to consume animal meat, he doesn't have to consume high amounts of carbohydrates, we want to increase high amounts of plant proteins in it. So that is this one. Okay, so second, whenever we are trying to do this, whenever we are trying to increase the amounts of proteins, plant based proteins in a food substance, there is a lubrication failure, because plant proteins, which I have already told you in the previous slides, that plant proteins come with extremely high amounts of astringency and lubrication failures. Okay? So whenever we try to do this, so tribology comes into play. You have to understand that how much amount of substance we can add into it. So whether any high amounts of protein can be added, whether any high amounts of sugars can be added, whether any high amounts of fats can be added, whether any high amounts of vitamins can be added, and then optimize all this. So nutritionally, like for example, the old people, old or geriatric people, they need very high concentration of proteins in order to maintain their muscle mass. Old people generally lose muscle mass. Okay, so they need high amounts of proteins. But the moment we try to incorporate very high amounts of proteins in the diet, it results in dryness, and old people lose capacity to swallow food also. So this is two opposite phenomenon. The more proteins, the more dryness. They should have less dryness. Okay, so these all things have to be taken care of. All these things have to be optimized. The dryness has to be less, swallowing has to be high. So that is why we need the study of tribology. We already discussed why do we need to study rheology. Now we are just studying that why do we need to understand tribology. Okay, so this will kind of understand, and then we will take a break. Okay, so in order to understand the tribology, as I said that the tribology relates to frictional properties, friction between two surfaces during mastication processes. So this can be mimicked, the tribology can be mimicked by putting a food sample in between a stationary phase and a rotating phase. Okay, so if I am here, only just a second. Okay, well, go back again. You just see this diagram here. So, this is an advanced this tribometer. I will go back again. So, do not focus on anything else, just focus on this diagram. This is an advanced tribometer which contains a bowl like structure, and in that bowl like structure you put your food material, and this bowl like structure acts as an oral cavity. It acts as a mouth. This mimics your mouth, oral cavity, and you see this part, this is a bearing. This is actually a bearing, circular in shape, very smooth. This acts as the tongue and the upper palate. Okay, so food material comes into contact with this bearing. This bearing rotates at different speeds and mimics the motion of your tongue. Okay, so this tribometer is an advanced version of a tribometer. We can have different kinds of tribometers, which will go after these slides. So what I wanted to emphasize here is that if you just take an example of this roller ball which keeps on rotating, mimicking the tongue, and any food material coming into contact with the disc, this lower part is usually stationary in nature, and the food material is kept in between the ball and the disc, we understand the frictional properties. How much amount of friction will happen when we do different kinds of motions, relative motions between the food materials and the ball bearings, that is actually the principle of tribometer, or that is actually the principle of tribology. What happens to the food materials when food material is consumed or food material is in our mouth? So the lubrication there, whether it remains for a pretty long period of time or whether the lubrication is gone, so that lubrication or friction properties between two surfaces which are in relative motion. So either one can be stationary or both can be in motion. So that is the principle of the tribology, or that is the principle of the tribometer. The way rheology is studied on a rheometer, tribology is studied on a tribometer. And rheometer is different, just a second. Rheometer is completely different and tribometer is completely different. Okay, they are two different equipments, but recently we have purchased a mini tribometer or an attachment only which can be attached to rheometer. Rheometer is a different equipment altogether which you have already seen, and then there is an equipment called tribometer. Okay, but recently the rheology or rheometer comes with an attachment which can also study tribology. So that is referred to as rheotribometer.
Okay. Sir, I want to ask one question regarding this tribology only. So that you have rheometer, and we have seen in the department as well, and tribology has a different you can say a tool or probe. It will be there,
I will just make
which can be connected to the rheometer.
Yes.
And just few minutes back you were telling about that there is a cavity like structure which will be mimicking like tongue. So is it a combination of e-tongue and texture analyzer or rheoanalyzer?
I will just make you understand. So if you have, I mean, you will understand it more, probably some people will not understand. So if we have to study the starch properties, those properties are, for example, called as pasting properties. So in pasting properties, we generally use an RVA, rapid viscoanalyzer, okay, in order to understand the viscosity profiles of starch, what happens to starch during cooking, what happens to starch during mixing, what happens to starch during temperature change, water, and blah blah blah. We use an RVA, altogether a different equipment. So but you must have seen that the rheometer also comes with an attachment called as starch cell. So if we attach this starch cell to rheometer, we can study all the properties that we study on RVA also. Okay.
Of starch
On
after gelation means we can do it on texture analyzer as well.
No, not texture analyzer.
No, to study the effect of temperature on the starch properties, we have to use RVA. But how it behaves after the gelation,
Exactly. If
we
you're
can
forming a
study
gel,
that
those
on
gel
texture
properties can be
also.
studied through
But you're
texture.
saying that we don't need texture, we don't need RVA, we can straightway study the impact of temperature as well as the pasting property on the
Pasting properties,
rheometer.
not that textural
It is the
texture
advanced
wala.
version.
Yes, it's an advanced version. So
Okay.
starch, this is called starch master. So this is an attachment which is attached to a rheometer. Okay. So similarly, so now if we have to study the rheological characteristics of any material, we will study it on a rheometer. But if we have to study the tribological properties of any food material, we will study it through tribometer. But our rheometer, which is an advanced rheometer, so it comes with an added attachment which mimics the same properties that we study on a tribometer. Okay. So probably you can just show them the attachment that we have for the tribometer, that we attach to rheometer. Otherwise, generally in practice, rheometer cannot give you the data related to tribometer, but we have a specialized attachment which can be attached to a tribometer in order to study tribology. Okay. The way rheometer cannot give you pasting properties of starch, but we have a specialized attachment which is usually called star cell. The moment we attach it to this rheometer, we can get different kinds of pasting properties of starch also. Please note now. So the way in case of a rheometer, if we have to do shear stress versus shear strain relationships, okay, in a rheometer. So similarly, we generally how much amount of friction is generated, friction factor, when sliding velocity and viscosity of any food product and the amount of the load. Load means how much amount of force is applied, how much amount of pressure is applied on a food substance. So this is varying, and how much amount of friction is generated in the food substance. The relationship between these two, okay, please note again, I think I don't have to write here, I am sorry. Oh, in case of rheometer, I will again go back. Simplest rheometric data is to draw the relationship between shear stress and shear strain, and we get different kinds of curves, whatever be. This is in rheology. Okay. In tribology, we get the data between friction coefficients, and these three are applied together: the velocity, changing velocity, the changing viscosity, and the load. These things are taken into consideration together. The relationship between these two is referred to as Tribic curve. So the output of a tribology is in the form of a Tribic curve. So wherever you get to see Tribic curve, just close your eyes and say that this is tribology data. Okay. So and then it will be the relationship between the frictional coefficients and velocity. Now again, going back, which I was discussing, which probably was a little bit boring, but you see that initially when we consume our food, it will be in contact with one surface. If this is a food material, it will be contact with the surface, and mild amount of saliva is in contact with the food substance. This is referred to as boundary lubrication. The first stage of consumption, whenever we are putting our food on our tongue and then it comes into contact with surfaces, it immediately will be coated with saliva, but not so much coated with saliva. Only one layer of the food substance will be coated with saliva. That is known as boundary lubrication. Okay, it has not been 100% lubricated. Second, the lubrication will go on increasing. This second stage is referred to as mixed lubrication. Lubrication will go on increasing. More surface of the food substance will be coated with saliva, and finally, when almost entire of the food substance is coated with saliva, that stage is referred to as hydrodynamic lubrication. So three stages of lubrication are there for any food substance or any other material also. First one, boundary lubrication, second one mixed lubrication, and third one hydrodynamic lubrication. You see that the friction coefficients are initially very high. That means saliva is not 100% coated with the food material, and then they go on decreasing, and then slightly increase. Okay, why that increases, I will just explain that in coming, why this actually happens, because there are a lot of other factors coming into play. Okay, so this is kind of an understanding of what happens during this Strebic curve. Okay, so in boundary regimes, the frictional coefficients are very high. So boundary lubrication means, now if you just close your eyes and just understand boundary lubrication, mixed lubrication, hydrodynamic lubrication. Boundary lubrication, starting processes, then mixed lubrication, kind of an in between phase of two extremities, and then final phases where we have a hydrodynamic lubrication. Initially the friction coefficients are very high, and then friction coefficients reduce, and then this thickness of the fluid also goes on increasing, and we have a lot of things coming into play. Okay, so let's just read that in coming slides. So here, see, now how exactly it gets affected. So shall we take 5 minutes or 10 minutes break and then start again? But I am not sure if anybody is here. Okay, so let's have this slide and then we will take a break. So in oral tribology, lot of things come into play. Number one, the type of food, interaction with saliva, and food, saliva, and oral surfaces. They all come into play to tell you how much any substance is smooth in nature, how much any substance is creamy in nature, how much any substance is rough in nature, or any substance is astringent in nature. For example, see, this oral tribology or tribology as such does not mostly depend upon the psychological factors, the way rheology is dependent upon it. Okay, so it generally depends upon our teeth, it generally depends upon our tongue, and it generally depends upon our salivary glands. That's it. Okay, so food lubricity or tribological stuff, it is generally dependent upon the food systems, the chemical properties of the food substance, the physical properties of the food substance, the stability of the food substances. That means whether any food substance has higher proteins, whether any food substance has more water solubility, whether any substance has less water solubility, whether it is it is hard in nature, whether it is soft in nature, whether it is a stable emulsion, whether it is unstable emulsion, or a lot of factors. And then it also depends upon our oral conditions. That means the conditions that are present in our mouth. Number one, what will be the temperature of the oral cavity, what will be the time that it stays in the oral cavity. So how much amount of pressure the tongue and the palate experience together, that also depends. And then saliva, how much amount of saliva is released. Some people have a problem that they don't have any saliva, so they have a natural dryness in their mouth. So what is the composition of the saliva, whether it is stimulated saliva or whether it is unstimulated saliva. Okay, so sometimes what happens is that before even taking any food product, before even you have taken any food product, if somebody tells you that okay, you want to taste pickle, you will already have a stimulated saliva in your mouth. But on the contrary, if somebody will tells you that just taste this rice spoon, after you have put it in your mouth, saliva gets secreted. Okay, so there are all these things that come into play. And then surfaces, whether the tongue surface is rough or speed of the tongue, the direction of the movement of the bolus or everything, all these things play. Food lubricity is dependent on all these things. So just take you through again. So before we go into tribological data, the frictional coefficients or frictional properties are studied using a tribometer. So either the tribometer is a friction tester apparatus, it just kind of tests the friction. So we put rolling bearings in a food material and it keeps on rotating, it just checks how much amount of friction is there. But the advanced one is this MTM. So friction tester, all ball on disc. So that means just a rotating wheel like, just imagine a wheel like stuff which keeps on rotating and it's passed through food materials and it just kind of gives frictional properties. or a ball on disc, a disc kind of, we have those marbles. It's kind of almost a similar marble like attachment, which just keeps on rotating, very smooth surface, and then these are just used for screening purposes, like they don't give you so much amount of reliable data. On the other hand, advanced setups like MTM machine or OTC is called as optical tribological configurations. This combines together different equipments along with the MTM machines. These are advanced ones. Okay, all these advanced ones. So either single, this is a double one. This MTM has single bearing or single balls. This is advanced one. This is also against advanced one. So these ones, they give you an idea of microstructure also, they give you an idea of lubrication also, and they give you an idea of sensory perception also. So these are the tribological configurations. So this one I will just, as of now, this is a kind of an homework or concluding from here. Is there any is there? So as of now, is there any difference between rheology and tribology now? Is there any difference between rheology and tribology? Is there any difference between rheology and tribology? Yes, there is a difference between rheology and tribology. So in rheology, for example, we are applying stress and strains, and then we are mimicking what happens to the food substances, they will flow. Okay. In tribology, different things are happening. We are applying load also, that means tongue and upper palate load, and then we are swallowing also, slipping also happens. So these the frictional properties as a result of load, as a result of slipping, as a result of being in the mouth. So these properties, they are usually in the form of lubrication processes. So there is a difference between rheology and tribology when it comes to the food substances. Okay. So we will take this slide and then we will take a break, just to make you understand what exactly saliva is all about. So again, going back to what I told you. So if we take, please note this one. This is something that we came to know after a long research. So tears, saliva, and synovial fluid. Natural lubricants. All three are natural lubricants. So tears also have proteins. Saliva also has proteins. Synovial fluid also has proteins. Okay. So tears have proteins, it's composed of proteins. Saliva also has proteins, and synovial fluid also has proteins, and all these proteins have mucins in them. Mucin is a type of protein. This is a very generalized statement. So mucin is a type of protein which is found in tears also, mucin is found in saliva also, and mucin is found in synovial fluid also. Okay. Please note, if you have a frictional relationship, so just kind of understanding what happens to the frictional coefficient when we are studying tears. Suppose the frictional coefficient of tears with as such, if we just use tears and study its frictional properties, the friction of tears initially is very low and it goes on decreasing like this. It has very low frictional coefficients. Okay. Contrary to that, if you remove mucin out of it, if you remove mucin out of tears, the frictional coefficients are like this. Frictional coefficients are very high. Okay. Same is true with saliva also, same is true with synovial fluid also. If you take out mucin, out of saliva, it will have very high frictional coefficients. So in other words, what I want to emphasize here is that the lubrication properties in these systems, is generally mucin driven. The lubrication properties in these systems is generally mucin driven. Okay. So saliva is composed of water, it is composed of solids, water 99%, more composition of the saliva is of water, while as solids are too less, 0.5%. And those solids, most important ones are enzymes and some of the organic substances like mucins. Okay. For example, amylase enzyme, maltase enzymes, lingual lipase, lysozyme, phosphatase, carbonic anhydrase, kallikrein. Kallikrein is a very specialized type of substance which is produced in liver, and it is generally responsible for protecting against flaking. Flaking means you will see some people have skin which is dry and rough, and they sometimes have that flaky skin. Okay, this kallikrein helps to prevent that. So this is an antimicrobial also. It has got other properties also. One of the properties that it helps, it's kind of an anti-flaking substance. So all these substances together are present in saliva. Okay. So, but if we talk about saliva in terms of the lubrication, please note that the lubrication in saliva is generally due to mucin, which I already told you. If you take away mucin out of saliva, if you take away mucin out of synovial fluid, if you take away mucin out of tears, you will see that its lubrication is not so good. Its frictional properties are very high. Okay. So this saliva, what it helps you, the interaction of saliva with food is this way. The interaction of saliva with food is this way. Number one, it coats the food and helps to break down. Okay. So when the food comes into contact with saliva, saliva coats the food substance and it helps us to break it down. And then testants. Testants means whatever substance is there in the food substance which is giving us the actual taste, those testants are transported from one place to another place and through saliva only, and then we can actually perceive the taste of the food substance through this saliva only. Okay. Then saliva also helps to change the rheological or colloidal properties. So for example, if you are consuming anything, so chocolates, there is a very great paper that came from University of Leeds which actually told that it is not just the fat content, it is not just the chocolate content, it is also the amount of the saliva and the coating of the saliva that tells us whether this chocolate is good or bad, or the mouthfeel of the chocolate also. So that paper actually came from University of Leeds only, and it was all over the world in almost every newspaper and magazines. So this saliva, when it comes into contact with food material, it changes the rheological properties also, it changes the texture also, it changes the fat related properties also. So then obviously when this saliva comes into contact with some other substances, the lubrication properties change. Please note it. This we have to study actually in the coming slides. So when we are consuming any food substance which contains some substances, say for example phenolic compounds, these phenolic compounds react with proteins. Proteins as in mucin. Mucin is a protein. This is what I was teaching you. So in the coming slides, everything will revolve around this one. So if you have not been attentive here, it will be difficult for you to understand the next slide. So lubrication, when there is complexation means mixing of two different substances. So when proteins, for example mucins, complex with other, for example substances, say for example polyphenols, there is a lubrication failure. So saliva is responsible for giving you the lubrication, and saliva might be responsible at times in breakdown of lubrication if it comes into contact with some other substances. Okay? And then aroma compounds, whether these some substances when they come into contact with saliva, they release different kinds of flavors and you just perceive it. Can we take a break for 5 minutes? Okay, so welcome back. So I mean, after a quick short discussion on whether rheology and tribology are same or different. So we'll go quickly to some of the case studies to make you understand that how tribology and rheology comes into play. So I think we just discussed about this, what is saliva and how saliva and food interact together. Okay. So as this I already told you. I mean the food saliva interactions come into play at different levels. So, one of the level is at structural level and another is at molecular level. So, at structural level, when food comes into contact with saliva, so it gets coated, it gets wet, food substance gets wet, it gets coated with this saliva, and smaller particles cluster together. So, they come together and lubrication starts. The moment this saliva comes into contact with the food material, it coats the food material, becomes wet in nature, food material becomes wet in nature, and smaller particles cluster together, they combine together and lubrication starts. And as I told you in the previous slide, that all the substances which are tastants, so that means wherever from the taste comes, all those they dilute, so they start getting dissolved and we start perceiving taste also simultaneously. And then this is very important that bolus formation happens. Bolus formation means bolus is it's a mixture of saliva and chewed food. Okay, when food comes into contact with saliva, we start chewing it, and then you start forming it in the form of a ball like stuff, and then the moment you start swallowing it, that's called as bolus. Okay, so this bolus formation is very, very important, and swallowing happens only after bolus formation. In elderly populations, or those who have some physiological problems, that means they are inherently having dry mouth, that disease is called as xerostomia. So, if somebody is naturally suffering from lubrication failure, that means his mouth is not secreting enough saliva, salivary glands obviously. So, that disease is referred to as xerostomia. Okay. And then when he is not able to swallow that food, that disease is called as dysphagia. Also, I have seen most of the people nowadays, they are studying on 3D printing of food substances for dysphagic patients without actually understanding tribology into it. So, we cannot understand dysphagia or dysphagic foods without having an understanding of tribology into it. Okay. So, this happens at structural level. When the saliva comes into contact with food materials, these all things happen at structural levels. But what happens at molecular levels? So, when saliva comes into contact with food materials, the destabilization happens, particles get broken down, and then it also has an effect on rheological properties. It has an effect on mouth feel sensations, and then hydrolysis of different kinds of macromolecules occurs. If there are proteins in our food substance, they start breaking down. If there are carbohydrates in our food substance, they start breaking down because of the action of different kinds of enzymes. Okay. So, flavor compounds which are present in the food substance, they start getting released. Different kinds of taste substances which are present in food substance, they start getting released. So, all these happen at molecular level. Then different kinds of complexizations which I will focus more on here. Please be attentive. So, if these salivary proteins which are mucin, salivary protein is mucin, when it comes into contact with food materials which contains astringent substances, for example, it contains catechins or it contains plant proteins. When it comes into contact with salivary proteins means mucin, okay, or let us say that it comes into contact with saliva. Saliva will try to do the same thing that it tries to do with the rest of the food substance. It will try to break it down, it will try to bind it with it, it will try to make bolus out of it, it will try to make it wet, it will try to reduce lubrication, it will try to coat it. Okay? But the moment saliva comes into contact with these astringent compounds, it forms different kinds of complexes. Please note, the moment saliva comes into contact with these substances, it forms different kinds of complexes. I will stop here because I have kept a purposefully a diagram in the next slides where I will show you what type of complexes are formed, for example. Okay. So, here when saliva comes into contact with these type of substances, so there is a breakdown of lubrication. How that happens, I will just show you. Let us skip this one. So, just keep it in, if you want, you can just take a, I was telling you that I have kept a link on sensory analysis. So, you can take a screenshot of this one, or you can just refer it later on also. I do not know why I kept it here, I should have been before. So, either you can visit this link and study different kinds of sensory evaluations. Sensory evaluations come with limitations, methodological limitations, bias, mouth cleansing, instrumental problems, and lot of stuff comes into play, whereas instrumental things do not have these type of issues. Now let us focus here. Let us first discuss about this one. This data is a light microscopy data, and this data is TEM data. TEM means transmission in electron. It's a kind of a, like we have simple microscopes which are used in this microbiological examinations. Then we have higher magnification, higher high-end equipments, scanning electron microscopy, FESMs. Then we have TEMs, we have AFMs. Lot of equipments are there. So on the left side, you see all these diagrams are from light microscopy. On the right hand side, they are all from TEM images. Okay. So all these clusters, these clusters, they are astringent compounds. They are astringent compounds. They are large, visible astringent compounds or astringent substances which are formed from protein tannin precipitates. Tannins are substances present in wine. This study is related to wines. Okay, what happens to the lubrication, what happens to the astringency when somebody tastes different kinds of wines. So for example, you see this is only with saliva. I will explain it. Saliva with one type of wine, saliva with another type of wine, saliva with another type of wine, saliva with another type of wine. So their friction coefficients are different. I will explain that. Okay, so when any person or this tannins come into contact with saliva, this saliva tannins make different kinds of precipitates. So similarly, on the right hand side, you can see different kinds of precipitates, saliva tannin precipitate, this complexes, sorry. So as soon as a person consumes any food product which contains astringent compounds, these astringent compounds come into contact with saliva and they make protein tannin, protein ABCD, whichever compound, astringent compound is there. They make different kinds of complexes, they make different kinds of precipitates, or you can say that they draw saliva out, they catch saliva. The work that saliva used to do normally, it used to coat the food material, it used to wet the food material, it used to make the bolus out of food material. Those all properties are lost because these compounds, for example, these tannins which are present in wine, they make different kinds of precipitates, they make different kinds of complexes with these with the saliva or the mucin. As I already told you that mucin is responsible for the lubrication. So this mucin makes different kinds of complex protein complexes with tannins, protein complexes with EGCG or any other astringent compounds, and the lubrication failure happens. Okay, so you understand this way that when plant proteins will also be consumed by human beings, when plant proteins are consumed by human beings, they also contain different kinds of phenolic compounds. Okay, inherently, naturally, they contain different kinds of plant proteins. Plant proteins contain different kinds of phenolic compounds which are astringent in nature. The astringency in plant proteins is because of different kinds of phenolic compounds. Okay, so those phenolic compounds get bound with saliva. They make different kinds of complex. They make different kinds of proteins, thus increasing the lubrication failure. Lubrication failure or dryness occurs in the mouth. So friction coefficients increase. Friction coefficients increase as astringency increases. The more the astringency, more friction will occur. So more tannins, more friction will occur. Total tannins, more astringency. So this is the relationship between whether astringency will be, if astringency will be more, friction will be more. If tannins will be more, friction will be more. If total tannins are more, astringency will be more. So this is kind of data from sensory evaluation correlated with data from oral processing, correlated with data from tribology. So if we draw the tribology that I showed you, Strybeck curve, that what will happen to friction coefficients when different comes up. You see this saliva, it has the lowest friction coefficients here. I can't see it 100%, but this is the lowest frictional coefficients here. Saliva alone has the lowest friction coefficients. When saliva comes into contact with different kinds of wines, friction coefficients increase. When it comes in contact with other type of wine, friction coefficient increase. When it comes in contact with other type of wine, more friction coefficients increase. So this will also tell you that different kinds of wines will have different kinds of polyphenols. Intensity might be more, therefore lubrication failures are more. Okay. So this is same what I told you in the previous slides. Yeah.
Can we always associate high astringency with low lubrication?
High. If there is high astringency, lubrication will be very, very less. Friction will be very, very high.
Yes. So it is always the same phenomenon.
Yes. Unless something happens, I will tell you what will happen. Let me just. This is same. Okay. Yeah, obviously, astringency is because of different kinds of compounds, polyphenolics in case of. Friction increases, yeah. And plant proteins, it's very, very difficult. I mean, it will increase the cost of production if we remove all those. I mean, if you do extra filtration processes for plant proteins, it will be extremely costly then. Animal proteins do not come with all these astringency factors. Plant proteins come with these astringency factors.
So means the presence of certain phytonutrients like alkaloids and other compounds, those are responsible for the astringency, and those are decreasing the.
They are responsible for lubrication failure.
Friction.
I will first go to one of the papers that if I'm kept it here. Okay. So one of the way outs that we kind of researched is that plant proteins, they inherently have very high astringency or they have very high friction coefficients, but from nature, I was already discussing this. If you see this paper, this one, if you study this paper, we opined here that we gave an opinion that in nature this saliva, it doesn't only have mucin proteins, or it doesn't have only proteins, it comes with glycated proteins. Okay, I will explain this. So tears, it doesn't only have proteins, it has glycated proteins. Okay. So we gave an opinion that whether the same phenomenon can be used for food systems also, and we can have ultra low lubrication coefficients. This saliva and tears, they have very, very low friction coefficients, very low friction coefficients because of the glycated proteins. Glycated proteins means proteins are combined with carbohydrates through different phenomenon. Okay, they are joined together. For example, one of the ways is to have Maillard reactions. Okay, glycation, proteins, polyphenol, proteins, carbohydrates. So different combinations can be used. So we have seen that if we want to use plant proteins in real world scenarios, so one of the way out is to use, for example, plant proteins which have been modified by doing glycation. In nature, it is there. Ultra low frictional coefficients, that means very high superior lubricity in tears, in synovial fluid, in saliva, is only because of glycated mucin. Mucin is not present as only mucin, it is present as glycated mucin. Okay, that gives it very high lubricity, that gives it very low friction coefficients. So we tried to find out, if we have a potato protein solution, what is the viscosity profile of that? Okay, you see here, viscosity profile of plant proteins at different shear rates. Then we tried to find out what is the viscosity profile of potato and pectin combined together. we tried to find out the viscosity of potato protein, we tried to find out the viscosity of pectin, we tried to find out the viscosity of potato protein and pectin at different ratios. And we found out that the as you said, that it was potato protein driven rather than pectin driven. Frictional coefficients or tendency to have very high viscosities was more protein driven. That means the more concentration of protein you added, the more this friction coefficients you are having, more lubrication failures you will have. So, this was the outcome of the study, that if we are having potato proteins, which most of the people do. So, we study the rheology of the proteins, we just forgot about tribology. Then we studied the rheology of potato proteins, we studied that this is the tribological data. This is the tribological data. So, we studied friction coefficients as entrainment speed. Entrainment speed means how faster that ball will rotate. PDMS, that's a PDMS surface. Those balls are PDMS surfaces, okay. So, they keep on rotating at different kinds of speeds. How much amount of friction occurs, you will see that as faster speeds occur, as more coating occurs, the friction coefficients will reduce. But we find out that the potato protein had a very high friction. Then obviously you see this potato protein is here only, blue. Potato protein is very high, and the moment you started combining potato proteins with pectins, it had lower friction coefficients. So, this was the outcome of the study, that more amount of proteins. So, that also tells you here, one of the study that I kept here is this. This is also I think from University of Leeds only. I am fluctuating here and there to make you understand more, this one. So, one of the studies that was done is that when we are incorporating different kinds of proteins in systems, for example, in case of juices, milk or other substances. So, if we are incorporating potato protein, if you are incorporating pea proteins, if you are incorporating soy protein isolates or like that. So, the moment we are increasing the concentrations of proteins, the more amount of friction coefficients come into play. More friction. Okay, less lubricity, that happens. So, some of the studies that you can study, I mean, if you really want to study more, I mean, I just don't want to go into too much details, because it will be little bit higher concepts for most of the students who are in their undergrads and postgrads, but these two papers can serve as kind of the starting materials, or kind of something that you can start with to understand more of the tribology of plant proteins. Okay, if this is the starting paper that came out of University of Leeds, and we tried to use potato proteins and pectin mixtures. And then this is how exactly what exactly influences the frictional properties, what exactly influences the tribological properties or any other substances when we are using plant proteins as such. So, now if we have the ideas, I think we will just stop after this, sorry, okay, 1 or 2 slides only, then we will stop. So, this we already discussed in the previous slides, that whether there is any whether rheology and tribology are same, they are not same. But there is a link between rheology and tribology, that rheology and tribology come into play interchangeable. I mean, when we are eating any food substance, when we are tasting any food substance, when we are trying to texturize, mimic any food substances, rheology and tribology are very important. Okay, and then if we have the ideas of rheology and tribology, both rheology and tribology, we can have newer kinds of foods in the market. So, for example, we have 3D printed foods for dysphagia patients. We can have better flow properties of the food substances, we can have better lubrication properties, we can have better taste of food substances, and nowadays people are also using fat replacers. I mean, as I told you, if the foods are being used for the purpose of healthy foods, we are removing fats out of it. The moment you remove fats out of it, that also will result in high friction, not because of astringent compounds. The moment you remove oil from the food substances, they will become more kind of harder in nature. Oil gives you softness. Okay, so when we are trying to use fat replacers, so some people are using proteins as fat replacers, some people are using starch as fat replacers. So their the fat replacing also has to take into account the rheological properties also and tribological properties also. And then, for example, if you are designing protein based emulsions or protein based meats or other substances, the microstructure of the food substances, the viscosity of the food substances, or smoothness of the food substances, all these applications can be easily understood if we have the better ideas of rheology and tribology. Okay. So this is emerging. What new can be done? Maybe incorporate AI and machine learning. You can have a lot of data from texture, data from rheology, data from tribology. Say for example, you have got 100 proteins. You have got 100 proteins. 100 proteins in the sense that plant proteins are, say for example 70, and then you have dairy proteins, say for example 10, then you have animal proteins, say for example 5, and you can do texture analysis, you can do rheological analysis, you can do tribological analysis, and then use AI and machine learning data to predict that if we use concentrations of this, if you use concentration of this, n number of combinations can be used, that you can have kind of, this is one of the research areas that we can do. And then as you were saying that probably we'll have robotic tongue, we can have different kinds of systems that will really mimic the saliva food interactions, which as of now is very, very difficult. So clean label, as I said, natural biopolymers can be used, new types of plant proteins can be used, new protein polysaccharide conjugates can be used. What I told you, this Maillard conjugates or combinations of plant proteins and carbohydrates, they are also called as conjugates. Plant protein conjugates can be used for developing better systems with better lubrication properties. Okay, and then different kinds of proteomics data, different kinds of studies can be done with tribological data in order to have better structure, in order to have better rheology, in order to have better tribology, in order to have better sensory properties, and all this can be used to have better consumer preferences. So this is a generalized rheometer that we have, and here in the lab we have a rheometer with tribology setup. So we can replace this setup with equipments that we have the tribological setup. So if you want to know more about our group, so you can follow us. We'll be happy to have you in our team for short visits, for research projects or anything. So do we have any questions? So one of the stars of our group is here, Devo. He is working on bean proteins. Okay, so he is doing something very fascinating. Okay, that's all for us. Thank you. Okay. So if you have not understood something, or something has been maybe out of context or whatever be, you can come to my office anytime, we can discuss it. Okay. So also great thanks to Dabu, he has helped a lot in making the presentations, and but we didn't present any data from your, anyway that was not so much related to tribology, right? Done. Okay. Chaliye, we'll catch up. Oof.